Welcome back to the SANS Channel podcast, where we discuss the coolest scientific papers and preprints of our day. If you're finding these deep dives into the world of science interesting, please do hit that like button and subscribe. It really helps us grow and bring you more of these conversations.
Absolutely. We appreciate your support as we explore some really fascinating and sometimes paradigm-shifting ideas coming out of current research.
Today, we're diving into a paper that really makes you reconsider something we thought we understood pretty well. Cancer. For decades, the prevailing view has been that cancer is fundamentally a genetic disease. A collection of bad mutations piling up in our cells.
That's right. This genetic paradigm, as it's often called, it's been the bedrock of cancer research, influencing everything from how we study the disease to how we develop treatments.
Okay, let's unpack this traditional understanding a bit. As the paper we're looking at lays out, the core of this idea is the somatic mutation theory, SMT, for short. Basically, the idea is that cancer starts with a single cell that picks up mutations in its DNA. These aren't the inherited ones, but changes that happen, during its lifetime.
And these acquired mutations, particularly what we call driver mutations, are thought to give that cell a growth advantage.
Right.
It starts dividing uncontrollably, leading to a whole population of genetically similar cancer cells, what we call clonal expansion.
So the picture has been, find these key driver mutations in a tumor, and if we can develop a drug to target those specific mutations, well, we can effectively shut down the cancer.
Exactly. And this drove huge efforts in genome sequencing, the whole field of precision oncology.
Sequence the tumor, find the genetic culprits.
And then go after them with targeted therapies. It sounds very logical, doesn't it?
It really does. But have you ever paused and wondered why, with all this focus and these sophisticated tools, we still face so many challenges? Why do cancers become resistant to treatment? Why does relapse happen so often?
That's the central question, really, that this emerging perspective grapples with.
Yeah. This paper argues that perhaps our laser focus on genetic mutations as the primary cause might be obscuring other crucial aspects of cancer development.
Things maybe we haven't paid enough attention to.
Exactly. They suggest we might need to broaden our view, maybe even revisit some older ideas from biology that look beyond just the DNA sequence. This deep dive is all about exploring these challenges to the traditional genetic view and how considering things like cell state and the tissue environment might change how you think about cancer.
So the paper kicks off by outlining what they call cracks in the genetic paradigm. Essentially, 10 observations from recent research that the traditional mutation-centric view struggles to fully explain.
Okay, 10 paradoxes. Let's hear them.
Well, the first one is quite striking.
Yeah.
There are many cancers that display very few or even no consistently recurring genetic mutations, yet they clearly exhibit malignant behavior.
So they act like cancer, but without the expected genetic drivers.
Precisely. Think of cancers like rhabdomyosarcoma or ependymoma. If specific driver mutations are the fundamental cause, how do these cancers arise and persist without them?
That's a really key point. It suggests having a mutation we associate with cancer isn't always a prerequisite.
Not always necessary, no. And conversely, the paper also notes that the pattern of mutations, the mutational signatures we see in cancers caused by known chemical carcinogens, can look identical to those in sporadic cases of the same cancer.
So just knowing the type of DNA damage doesn't necessarily tell us the whole story of how that cancer actually got started.
Exactly. Then the second paradox they highlight is, well, it feels like nearly every gene in the genome has, at some point, been implicated in cancer.
Like the list just keeps growing.
Yeah, it's become so broad that the idea of a few specific driver genes kind of loses some of its distinctiveness. If almost any gene can be associated with cancer sometimes, does that mean they're all equally important drivers? Or maybe?
Or maybe it points to something else underlying it all.
Could be. Which leads us to the third paradox, which I find particularly interesting.
Okay.
Normal, healthy tissues often contain a surprising number of mutations, including some that we would typically classify as oncogenic, capable of driving cancer.
Wait, so healthy tissue can have cancer mutations?
Yep. And this is even seen in individuals with inherited cancer syndromes who carry these supposedly cancer-causing mutations in every single cell from birth, yet they don't necessarily develop cancer in every tissue, or sometimes not at all for a long time.
Wow. That really underscores the idea that just having a mutation linked to cancer isn't enough to actually cause the disease.
It seems the normal tissue environment must have ways to suppress or compensate for these mutations. Context is crucial.
Absolutely crucial. Okay, paradox 4.
Paradox 4 looks at those indolent or slow-growing and potentially pre-malignant lesions, things like your average skin mole, or Barrett's esophagus, or DCIS in the breast.
Right, things that might become cancer but often don't.
Exactly. These often harbor what we consider driver mutations, yet the vast majority of them never progress to invasive cancer.
So the mutation is there, but it's not doing much.
It seems so. And interestingly, when they do progress, the resulting tumors are often genetically distinct from that initial lesion. It suggests something called field cancerization.
Field cancerization, like the whole area is somehow primed.
Kind of like that, yeah. Rather than just one mutated clone taking over step by step, it challenges that simple linear progression model.
So these mutations can sit there for years, decades even, without causing trouble until maybe the tissue conditions change.
That seems to be the implication. The mutation might be necessary, but not sufficient without the right context or other triggers.
Okay, number 5, ionizing radiation. We know it causes DNA damage.
Right. But contrary to what you might expect, if cancer were solely driven by immediate mutations, radiation doesn't typically trigger a rapid expansion of a single mutated clone right away.
It doesn't. What happens instead?
Instead, we often see multiple different clones growing, but later on after several cell divisions. This suggests the initial effects of radiation leading to cancer might be non-genetic.
Like cellular stress or changes in the local environment?
Possibly, yeah. Things that then promote genomic instability down the line, rather than the radiation directly creating the one magic cancer mutation instantly.
So the link between DNA damage and cancer isn't always a straight line.
Doesn't seem to be. Okay, number 6 gets into the idea of positive selection, a key concept in Darwinian evolution.
Right, the survival of the fittest cancer cell.
The genetic paradigm assumes that tumor cells with oncogenic mutations should have a fitness advantage, letting them outcompete normal cells. But evidence for these widespread, hard selective sweeps, where one single superfit mutant clone takes over the whole tumor, is surprisingly limited.
Really? So it's not always one winner takes all?
Often not. Instead, we see a lot of what's called neutral drift, where mutations that don't necessarily provide an advantage can become common just by chance, especially when the population is small.
So just because a mutation is common doesn't mean it's a driver?
Not necessarily. This complicates the idea that every mutation we find is a good target for precision medicine, if many are just passengers.
Targeting them wouldn't do much.
Right. The clearest examples of positive selection are usually in drug resistance, where a specific mutation in the drug target does provide a clear survival advantage when the drug is present. But even that isn't the whole story of cancer development.
Okay, so selection isn't as straightforward as we thought. What's paradox 7?
Paradox 7 looks at the sheer genetic complexity within tumors, what we call intratumor heterogeneity.
All the different genetic variations inside a single tumor.
Exactly. Lineage tracing studies, which let us follow the ancestry of cancer cells, reveal really intricate nonlinear patterns of clonal evolution. We see branching parallel evolution where different groups independently get similar mutations.
It sounds messy.
It is, and you even see clones that seem to disappear and then reappear when the cancer comes back after treatment.
Whoa, that doesn't fit the simple step-by-step model at all.
Not neatly, no. This high level of genetic diversity suggests tumors are more like dynamic ecosystems with lots of interacting players, not just one dominant clone marching forward.
An ecosystem. I like that analogy.
Yeah.
Okay, number 8.
Number 8 shifts focus from genetic heterogeneity to non-genetic phenotypic heterogeneity.
So differences in behavior even if the genes are the same.
Precisely. Even within a population of cancer cells that are genetically identical clones, we see huge variation in their gene expression and, consequently, their behavior. Single-cell transcriptomics lets us see this.
And it's not just random noise.
Doesn't look like it. It reflects real flexibility where cells with the same DNA can exist in different states. We even see mutated cells whose gene activity looks just like normal cells.
So genotype doesn't perfectly predict phenotype.
Not always. And these states can switch dynamically. Even progenitor cells with cancer mutations can sometimes still differentiate normally. And if you take genetically identical cancer cells, they can respond very differently to the same drug.
That flexibility seems really important.
It fundamentally challenges the idea that the DNA sequence is the sole determinant of cell behavior. Gene regulation and environment are key.
Okay, almost there.
Paradox 9. Paradox 9 introduces the concept of persister cells. These are alternative states cancer cells can adopt that let them survive treatment without any underlying genetic changes.
Survive treatment without new mutations. How?
They seem to enter a sort of reversible drug-tolerant state, be dormant-like. So a tumor might seem to develop resistance, but it's not necessarily because a new resistance mutation was selected.
It could be cells just hiding out in this persister state.
Exactly. And crucially, this state can be temporary and reversible. Once the drug pressure is off, they might wake up again.
That really goes against the grain of the SMT, doesn't it? Which emphasizes permanent genetic changes for resistance.
It does. Persister states are a fascinating example of non-genetic adaptation, and the reversibility is a big problem for the standard SMT logic.
Okay, last one. Paradox 10.
Paradox 10 builds on this resistance idea. It suggests that resistance, including these persister states, can actually be induced by the treatment itself.
Induced, not just selected for.
Right. Rather than just passively selecting pre-existing resistant mutants, the therapy stress might actively push cells into resistant states.
Like a direct response to the environment, almost Lamarckian.
The paper hints at that comparison, yeah. Stress can trigger cells to become more stem-like and drug-resistant through active signaling pathways, not just through passive selection of rare genetic mutants.
So our treatments could, in some cases, be inadvertently creating resistance non-genetically.
It's a possibility that needs serious consideration. It means strategies against resistance need to look beyond just finding resistance mutations.
Wow. Okay, so those 10 paradoxes together.
Yeah.
They really paint a picture where the purely genetic explanation has some significant gaps.
Significant gaps, yes. Which naturally leads us to question the somatic Darwinian evolution corollary that's so central to the genetic view.
The idea that random mutations confer fitness lead to clonal expansion, the fittest clone takes over.
That whole narrative, the paper argues this idea is under increasing scrutiny.
It seems like it would have to be, given those paradoxes.
Absolutely. While Darwinian evolution almost certainly plays a role, particularly in therapy resistance, where a mutation gives a clear advantage against a specific drug.
Right, that makes sense.
The evidence for it driving the initial development of all the complex hallmarks of cancer through these hard selective sweeps is actually quite limited.
We already talked about the heterogeneity, too many clones coexisting for one to have simply taken over completely.
Exactly. And remember, the neutral drift point mutations can become common just by chance, not necessarily because they're providing a big advantage.
So we can't assume every common mutation is a driver?
We shouldn't know. Plus, you have all these complex interactions happening within the tumor ecosystem.
Between different cancer subclones. And with the normal cells around them.
Yes, the tumor microenvironment. These interactions between subclones and between cancer cells and the stroma, they complicate the simple picture of one clone just outcompeting others based purely on its own mutations.
The ecosystem idea again. Maybe different clones cooperate.
That's a growing area of research. Maybe one subclone is better at invading another handles immune evasion, a kind of division of labor. Yeah. It's more complex than simple competition.
So if the simple neo-Darwinian model isn't the full story, what other evolutionary or ecological ideas might apply here?
Well, the paper mentions a few possibilities drawn from broader biology, things like punctuated equilibrium.
Long periods of stability, then rapid change.
Yeah, that might fit the pattern of slow growth followed by sudden aggression we sometimes see. Also, group selection focusing on the fitness of the group, not just the individual cell.
Relevant for cooperating clones.
Could be. Niche construction, how tumors modify their environment, recruiting blood vessels, suppressing immunity, that definitely happens.
And structuralism. Physical constraints, shaping things.
Right, the idea that not everything is solely down to selection. Physical and chemical laws also constrain possibilities, and of course phenotypic plasticity.
That ability to change behavior without changing genes. How does that fit evolutionarily?
There are theories suggesting this non-genetic flexibility can actually guide or influence the path of later genetic evolution, things like the Baldwin effect or genetic assimilation. So plasticity isn't separate from evolution, it can interact with it.
This is really shifting my thinking. If it's not just about genes and simple selection, what are these alternative views? The paper talks about cancer as a disease of gene regulation and cell state.
Yes, this is a fundamental shift. The idea is that cells with the exact same DNA can be incredibly different, like a neuron versus a skin cell.
Because different genes are turned on or off.
Exactly. It's about the pattern of gene activity across the whole genome, and these patterns are controlled by gene regulatory networks, GRNs.
The intricate wiring diagram inside the cell.
Sort of, yeah. These networks can settle into stable configurations, stable patterns of gene activity called attractor states. Think of a landscape with valleys. Each valley is a stable cell state, like skin cell or liver cell.
Okay, I can picture that landscape. Where does cancer fit in?
The theory suggests there might be many more potential valleys, more attractor states in that landscape than are normally used by healthy adult cells.
Latent states.
Latent, unused states. And some of these might correspond to malignant phenotypes, cancer attractors. Carcinogenesis, then, could be seen as a cell accidentally falling into one of these undesirable latent valleys.
Wow. So it's not creating something totally new via mutation, but accessing a pre-existing hidden program. The paper mentioned atavism.
Yes, the idea that cancer might be a throwback to more primitive, maybe single-celled organism behaviors, more selfish programs that are usually suppressed in multicellular life but are still somehow encoded.
Or maybe cells get stuck in an early developmental state. Immature.
That's another related idea. They fail to reach a mature, differentiated state and retain these more proliferative, less specialized characteristics.
And what pushes the cell into one of these bad valleys, these cancer attractors?
Various kinds of stress, the paper suggests. Even non-genetic stress could destabilize the gene expression patterns, making the network wobbly.
Like bumping the ball out of its normal valley.
Exactly. Making it easier to transition into a nearby but undesirable cancer attractor state. It's a different way to think about initiation.
So how do mutations fit into this picture? Are they irrelevant?
Not irrelevant, but maybe their role is more indirect. Instead of directly causing the cancer phenotype, maybe mutations subtly tweak the GRN wiring.
They change the landscape.
They might gently reshape that epigenetic landscape, maybe lowering the hills between valleys, making it easier to slip into a cancer attractor. It's a less direct, more probabilistic effect than the traditional gene X causes cancer Y view.
So most mutations don't cause a catastrophe, but they might increase the chance of transitioning to a bad state.
That's the concept. It challenges the simple one-to-one mapping between genotype and phenotype that underlies both the classic genetic paradigm and traditional neo-Darwinism.
What about big genomic changes, like aneuploidy?
The paper suggests those large-scale alterations might represent more fundamental generic attractor states that the whole system can fall into when severely dysregulated. Maybe even related to basic life cycle programs like dormancy or gamete formation, reactivated inappropriately.
And genomic instability, the high mutation rate in cancers, cause or effect.
This view leans towards it being more of a consequence. The abnormal state of being in a cancer attractor might itself lead to problems with DNA replication and repair, causing mutations to pile up. The idea that cancers beget mutations.
That flips the script. Okay, there's another big idea in the paper. Cancer as a tissue-based disease, the tissue organization field theory.
Yes, TOFT. This directly challenges the cancer as a disease of the cell focus. It argues the tissue context is paramount.
Is there evidence for that?
There's some classic experimental evidence, like taking liver cancer cells, if you inject them into the liver, they form tumors. But if you disperse those same cells throughout the liver via the spleen, they can actually get normalized by the healthy tissue environment.
The same cells behave differently depending on the tissue context.
Exactly. It suggests being a cancer cell isn't just an intrinsic property, but depends heavily on the surrounding tissue field.
So what are the principles of this tissue organization field theory?
Two main ideas. First, the default state of cells is proliferation, variation, and movement. What's remarkable isn't that cancer cells proliferate, but that normal cells don't most of the time.
So tissues normally impose constraints.
Right. Second principle. Cancer happens when these tissue-level organizational constraints break down, freeing cells to revert to that default proliferative, motile state.
It's a failure of tissue organization, not necessarily a cell going rogue because of its own internal mutations first.
That's the core idea, and it puts huge emphasis on the stroma, the tumor microenvironment. It's not just passive scaffolding. Well, experiments show that hitting the stroma with carcinogens, even if the epithelial cells aren't directly hit, can initiate cancer. The stroma directs cell behavior during development, and a healthy stroma can suppress tumor formation.
So the environment can be the primary target.
In some cases, potentially yes. And messing with the stroma isn't always good. Targeting certain fibroblasts in pancreatic cancer sometimes made things worse, paradoxically.
Wow, it's complex.
Very. And the theory also highlights physical factors. Chronic wounding, mechanical stress, they can drive carcinogenesis without direct DNA damage, pointing to the importance of tissue-level forces.
This sounds like an organicist perspective, looking at the whole system, the interdependencies, feedback loops.
Exactly. It sees the organism and its tissues as integrated systems where molecules, cells, and physical forces all interact in complex, often circular ways. This makes finding a single molecular target a magic bullet much harder.
Okay, so with all these alternative ideas and paradoxes, how is the traditional genetic paradigm held on? How does it try to explain these inconsistencies?
The paper suggests a common approach is adding auxiliary hypotheses. So, keep the core idea that mutations initiate cancer, but then add layers like, metabolism is altered, or the stroma plays a role, or the immune system is involved.
Patching the theory.
Kind of like patching it, yeah. Adding complexity to explain new data without fundamentally changing the core assumption about genetic initiation.
What about the initiator-promoter model? Does that fit?
That model tried to account for non-mutagenic carcinogens by proposing an initial mutation initiator, followed by a non-genetic stimulus promoter. But experiments showing the reverse order can also cause tumors kind of muddy those waters, suggesting a more complex interplay.
And epigenetics. Just changing gene expression without mutation.
Epigenetics is definitely important for cell states, but the paper argues it's probably not the whole story for the stable, enduring changes in cancer by itself. You likely need the underlying gene regulatory network dynamics, the attractor states, to maintain those changes long term. Epigenetic marks might reinforce a state, but the GRN structure defines the possible states.
It sounds like the genetic paradigm, maybe because it's become so broad, is hard to actually disprove.
That's a point the authors make. It might have become somewhat vague, allowing inconsistencies to be explained away with these add-on hypotheses, rather than forcing a fundamental rethink or looking for a more unified theory based on first principles.
They quote Darwin, don't they? About observation needing a theory.
Yes. Observation must be for or against some view if it is to be of any service. The paper calls for a more rigorous, theory-driven approach to understanding carcinogenesis based on fundamental biology, not just cataloging mutations and altered pathways.
Maybe the diminishing returns from just sequencing more and more tumors will be the pivot point.
Could be. When the mutation databases stop yielding easy answers, maybe the field will more broadly embrace these alternative, perhaps more systemic perspectives.
So, wrapping this up, what's the main takeaway here?
The key message is that the traditional genetic paradigm, while incredibly useful, is facing serious challenges. Accumulating evidence points strongly towards cell state dynamics, driven by gene regulatory networks, and the critical role of tissue organization being potentially primary drivers of cancer.
So for you, our listener, thinking about this gives you a much more nuanced view, doesn't it? It's not just a disease of bad genes happening in isolation.
Exactly. It's about the interplay between genes, gene regulation, the cell's environment, the tissue context.
Yeah.
It's much more interconnected.
This has been a really fascinating deep dive. It definitely leaves me with a final provocative thought for everyone listening. If cancer is, at least partly, about unleashing these latent cell programs or a breakdown in tissue control, well, how should that change how we try to prevent or treat it? Do we keep focusing mainly on targeting specific mutations? Or should we be thinking more radically, maybe about restoring normal tissue environments? Or finding ways to nudge cells back into healthy attractor states? What if that was the goal?
It's a profound question, and it definitely opens up a whole new landscape of possibilities for how we might approach cancer in the future.
Really makes you think. Please don't forget to like the video and subscribe if you want more content like this. We really appreciate you joining us for this deep dive into the evolving understanding of cancer.